Hey guys, my name is Abhishek and welcome back to MBBS channel. Today we are going to read about type 4 hypersensitivity reaction, that is otherwise known as cell-mediated hypersensitivity reaction. Till now we have discussed type 1, type 2, type 3 hypersensitivity reaction. Now we are going to discuss about type 4. So let's start our today's topic. So friends, we know that from type 1, type 2, type 3 reactions, we already read that it is mediated by the antibody. Like in type 1 is mediated by IgE antibody, type 2 is mediated by IgG and IgM, and similarly type 3 is mediated by IgG and IgM. So they have difference according with their mechanism of actions of pathogenesis. But in this type 4 hypersensitivity, it is completely different from the type 1, 2, 3. How? Because this hypersensitivity reaction is mediated by a particular cell, that is T lymphocytes. That is the reason why it is known as cell mediated. It is not mediated by any kind of antibody. Neither it takes help from the B lymphocytes. It only occurs or only mediated by the T lymphocytes, that is Th1 cell. We know from immunology that there are 4 kinds of T lymphocytic cells. What are these 4 T lymphocytic cells? It can be T helper cell, it can be T cytotoxic killer cells, and it can be T lymphokine producer cell, and it can be T suppressor cell. But in this specific cell mediated hypersensitivity, what happens? This T helper cell, and more specifically the T helper 1 cell, is going to mediate or start the process of hypersensitivity, and then it's going to result in damage of the host cells and tissue due to activation of specific effector cell. What are these effector cells? It can be macrophages, it can be natural killer cell, and it can be neutrophils. So the T lymphocyte, that is Th1, is going to activate these effector cells, macrophage, neutrophils, and natural killer cells. And this cell mediated reaction comes under the specific, that is delayed type of hypersensitivity. It comes under the specific classification, that is delayed type of hypersensitivity, and in short abbreviation it is known as DTH. So it is the hallmark of the cellular reaction. I am going to say the reason that why it's coming under the delayed type of hypersensitivity. While I am going to explain the mechanism, on that very moment I am going to explain that why it is known as delayed type of hypersensitivity. So next comes the antigen. As we have read the different types of antigens of different type of hypersensitivity reactions, so accordingly here also there will be presence of different kinds of antigens. Like say for an example, here the antigens we have divided according to 3 kinds. What are these 3 kinds? First one is the foreign agent. Like if there will be any kind of external substance that can alter the self antigen, then we will be saying that yes, this foreign agent will be a antigen for our body. Let's say for an example, when we are grafting or transplanting an organ, what happens? The cells or the host particular cells that are present in the organs or tissue will be acting as an antigen, and that can produce a delayed type of hypersensitivity reaction in our body. And another example, there is a plant, that is ivy poisoning plant, and when we come in contact with its leaf contains a particular chemical that is known as urushiol, and that urushiol can alter the skin protein. Okay, so when it's going to alter the skin protein, then we are going to see the delayed kind of hypersensitivity in this condition, that is contact dermatitis. Next antigen that we can see is autoantigen. According to the auto means self. So the self antigens can recognized by the autoreactive T cell. And when this condition actually occurs, this occurs due to failure of self-tolerance mechanism. It means when our body cannot recognize self antigen and extra or external antigen, then on that very condition we are going to develop these autoantigens, and it occurs due to failure or disorder of lymphoid organs. In case of this, it will be thymus, and here the thymus will be going to produce more number of T cytotoxic or T killer cells. That is the reason why it is written auto-reactive T cell. And we can see this condition in the delayed type of hypersensitivity, that is autoallergic condition. Next type of antigen comes that the antigens that are derived from the intracellular pathogens. What do you mean by this? There will be presence of pathogens and intracellularly the antigens will be derived from them. What do you mean by this? There are some kinds of microsomes or microorganisms that can be present in our body for a long period of time, and they can be present in latent form because they have the ability to resist the immunological clearance mechanisms, that they cannot be destroyed by our immune system, and they used to sustain for a very long period of time. That these three reasons is going to help them to produce the long term or prolonged infection in human beings. So let's see an example of this. Example of this is Mycobacterium tuberculosis. The Mycobacterium tuberculosis can persist in two stages. It can persist in latent stage and also in active stage. When a person is having latent stage of the Mycobacterium tuberculosis, then the person will be having less or no clinical manifestation at all, but there will be prolonged infection. So how we are going to see the type 4 hypersensitivity reaction in case of this? If we are going to diagnose the tuberculosis, whether it's present or not, through the tuberculin skin test or the Mantoux test, then we can understand that this reaction will proceed in this cell mediated hypersensitivity reaction. Okay, so these are the antigens that's going to trigger these kinds of hypersensitivity reaction. Next comes that when these antigens are present, then they should have certain kinds of mechanisms to being presented to the T cells. Yes, the antigen will be presented by the antigen presenting cells, like under the like the cells that are present under the skins or the mucous membranes. Let's say for an examples, the antigen presenting cells can be the dendritic cells, it can be Langerhans cells, it can be macrophages. So they are lining under the skin or the mucous membranes. So what is their function? Antigen presenting cell states that they are going to present the antigens to the next T cells, and according to their presentation or way of presenting, they are going to activate two kinds of T cells. What are they? If they are going to present the antigen peptide with MHC2 complex. MHC means major histocompatibility complex. So if they are going to present the antigen of peptide with peptide MHC2 complex, then it's going to form the CD4 T cell, and when it's going to activate the CD4 T cell, then it's going to proliferate and differentiate into Th1 cell, the T helper 1 cell. I want to make you remember one thing, that there are also Th2 cell that we have already read in type 1 hypersensitivity. Okay. Next comes that if the antigen presenting cell is going to present the antigen peptide with MHC1 complex, then it will be activating the CD8 plus T cell, which is exclusively rare condition. And if the CD4 T cell is going to be activated, then it's going to form or differentiate, proliferate into T effector cell. So this antigen will be presented by antigen presenting cell, and it will be forming these kinds of T cells. So this T cell is going to mediate the process. Let's see how the process is being mediated by the T cells. If we are going to consider the actual mechanism of the cell-mediated hypersensitivity reaction, this reaction has been divided into first and second stage. First will be the sensitization stage and second one will be the effector stage. Firstly, when there is sensitization, sensitization means the T cell will be sensitized, and when it will be sensitized, when there will be primary contact for the first time, when the antigen will be exposed to our body, on that very moment the T cells are going to be sensitized. Not on that moment, I mean to say that the sensitization occurs on the very first time exposure. Okay, so the T cells are going to be sensitized and the antigen specific memory T cells are going to be produced. What happens in this sensitization? When the antigen is going to be exposed for the first time, then the T cells are going to be sensitized. It means the T cells are going to recognize the type of antigen or what are the type of peptides that will be presented by the antigen presenting cells. Okay, so they are going to recognize it, and after the recognition process, they are going or they will try to memorize that by proliferating and forming the T memory cells or antigen specific memory T cells. That is the reason why in the sensitization phase there will be production of this memory T cells. Okay, and this sensitization process takes place for 7 to 10 days. So this is the reason why, as it's taking 7 to 10 days for the process of sensitization of a T cell. For first time sensitization of T cells, as it's taking about 7 to 10 days, that is the reason why it is known as delayed type of hypersensitivity. Type 1, Type 2, Type 3 comes under immediate type of hypersensitivity because the clinical manifestations are shown within 24 hours or within 48 hours. But we are going to see the first clinical manifestation on first exposure of sensitization stage, that is 7 to 10 days. That is the reason why it's known as delayed type of hypersensitivity. So what happens? Let's see this diagram. In this diagram, I have drawn this diagram to explain the sensitization stage. You can see here that upon first time exposure of the antigen, and what is the antigen here? I have taken example of this ivy poison plant. I have taken the example of the leaf of ivy poison plant. So what happens? It has the chemicals, that is urushiol. So urushiol when comes in contact with our skin, this black cells are nothing but our skin cells. Okay, it is the epidermis. So when it comes in our contact with the epidermis, it's going to affect the epidermis. How it's going to affect? It's going to penetrate the cell, and it also going to alter the cell proteins. These red structures that you can see, these are nothing but the cell proteins. So when it comes in contact with the cell, it's going to penetrate and alter the skin cells, and it's going to alter and penetrate. So after alteration, it can form like this structure, or it can also come selfly. So if it comes selfly, it is going to attach to the antigen presenting cell, or if it's coming through this whole composite manner, then it's also going to be attached to the antigen presenting cell. So here the antigen presenting cells may be dendritic cells or Langerhans cells. As it's a skin, so I am saying it's as dendritic cells. So the dendritic cells have specific functions. It's going to receive the antigens what's coming. So what will be done inside this? It's going to form a determinant form of the antigen or modifying the antigen into a peptide form. So that process is known as processing, and it's going to express that modified determinant form on its cell membrane, and that process is known as expression. So it's going to function process and express, processing and expressing the antigen and forming a peptide MHC2 complex. So as I have already said that according to the type of peptide presentation, there will be stimulation of the receptors, or there will be stimulation or activation of different kinds of T cells. So here in this case, it's going to activate the CD4 T cells. Also, along with this presentation of peptide MHC2 complex, as you can see, this is the antigen part, and it also going to produce some kinds of cytokines. What are these cytokines? It can be interleukin 6 and interleukin 12. So this activation of interleukin 6 and interleukin 12 is going to activate the CD4 plus T cell. I have written here one term, that is naive T cell. What do you mean by naive T cell? Listen, when it's coming exposed, or when it is exposing for the very first time, then our T cell never recognizes that what kind of antigen is it. So that is the reason why it is immature T cell or uninformed T cell. So it doesn't have any kind of experience to this particular antigen. That is the reason why it's the naive T cell for the very first time exposure. So upon activation of this CD4 T cell, what it's going to function? It's going to proliferate and differentiate into what type of cell? It's going to proliferate into Th1 cell. So there will be formation of Th1 cell, T helper 1 cell. So and with memory T cell, because as I have said, they are recognized, or they have recognized, and they need to memorize it. So they are produced in the, so they will be producing the memory T cells. So now in the next step of this sensitization, what happens? The Th1 cell is going back to the site of antigen. It is the site of entry of antigen, and it is the site of lymph nodes where the T lymphocytes will be present. So the Th cell will be going back to the site of entry of antigen. So in the lymph nodes, the process of activation of CD4 T cells or the naive T cells takes place, and proliferation of Th1 cells takes place. After that proliferation, the mobilization of Th1 cells back to the site of antigen entry. So this whole process of activation, proliferation and mobilization takes around 7 to 10 days. That is the reason why this sensitization phase of the hypersensitivity reaction comes under the delayed type of hypersensitivity. Next we are going to see the effector stage. What actually happens in this effector stage? Listen, this effector stage only occurs when there will be secondary contact or second time exposure to the same antigen. And in that effector stage, the conclusion will be the cell destruction, or the host tissue will be damaged. And it occurs between 1 to 2 days after the exposure. So it occurs more than 48 hours. So it occurs after 48 hours. So what happens when there will be second time exposure? Yes, this after the second time exposure, this whole entering process of the antigens will be recognized by this memory T cells, and this memory T cells is going to activate the Th1 cell. Along with this process and the memory T cells, the Th1 cell is going to be activated, and as the Th1 cell is going back to the site of antigen entry, back to this region, then what will happen? It will be releasing the interleukin 2, interleukin 3, interferon gamma, tissue necrotizing factor beta. So this Th1 cell is going to produce the cytokines, and these cytokines will be attracting the monocyte. The monocytes are present in the blood vessels. Okay, so they will be attracted to this site of entry, or the site of antigens. So these are the tissue or cell sites. Okay, and these are the blood vessels. So from the blood vessels, the monocyte will be attracted to this region, and when the monocyte is going to travel from the blood vessels to the tissue region, then it's going to differentiate it into a form that is known as macrophage. Then the monocyte is being differentiated into the macrophage, and this macrophage will be activated due to these cytokines that are present and that are secreted by the Th1 cell. So with the process of activation of the macrophage, it is going to also produce the cytokines and more chemokines that is also going to attract or invite more amount of this effector cells, and these effector cells like neutrophils and natural killer cells. These effectors and natural killer cells are going to produce the inflammatory mediators, and the inflammatory mediators will result in the non-specific destruction of the host cell, and finally the tissue will be damaged. So from this we understood that the tissue damage mostly occurs due to more efficient phagocytic activity by the macrophage and non-specific destruction of the host cell by these neutrophils and natural killer cells. So from the beginning, as I have said, that this Th1 cell is the primary mediator of this process, and the effector cells are the macrophage, natural killer cells, and the neutrophils. Okay, so this was all about type 4 hypersensitivity reaction. If you find this video helpful for you, please do like, share and subscribe to my channel. Thank you so much for watching my video.